for recalcitrant seeds, but I'm also gonna be talking a little bit about cryopreservation options for orthodox seeds that are short-lived or for other type of seeds that may present difficulties during storage in conventional seed banks. Well, I like to start this type of presentations a little bit reminding what probably all of you know, is that we are living in a moment of global biodiversity crisis where the last reports that have been written are suggesting that near a half of all known plant species in the world are threatened with extinction. And also, there are estimates, recent estimates that say that near 80% of all those plant species that have not already been described, so are unknown for science, they maybe also be threatened with extinction. That's a really big problem, not only for biodiversity conservation and ecosystems, natural ecosystems, but also for those useful plants for agriculture or for other uses that humans may need. When thinking on the proportion of these plant species that are threatened with extinctions, that are endangered, that possess seeds that cannot be conventionally banked in seed banks, which are these seeds called recalcitrant seeds, there is a third of all these species that, based on estimations made by Wise et al., that will probably have recalcitrant seeds, and the problem is that for the recalcitrant seeded species that we cannot bank in conventional seed banks, the only way to preserve them for the long term is through cryopreservation and through different sets of cryobiotechnologies. Just a little quick overview about what is a recalcitrant seed, although I imagine most of you know this. A recalcitrant seed, as I said, is a seed that cannot be preserved in conventional seed banks. It is a seed that have different physiological attributes. Mainly, it is desiccation and freezing sensitive, which means we kill the seed if we dry it below certain moisture threshold, which usually is set around 25-45% of the moisture content, which traduced in terms of water, grams of water per grams of dry weight of the tissue is about 0.2, 0.3. And because the seeds have that high amount of water, they cannot be stored in below 0 degrees Celsius because the water crystallize and those ice crystals kill the seed. Then we cannot bank them in conventional seed banks like orthodox seeds are banked. And apart from these two seed categories, based on these physiological attributes of desiccation, tolerance, sensitivity, and banking possibilities, there are other group of seeds that present some kind of features intermediate between the orthodox and recalcitrant seed limits. For example, they can be dry, but not up to those really low water contents that orthodox seeds can be dried, or they can be dry, but then when stored below 0, they present freezing sensitivity and they are killed by low temperatures, not by the drying, or they are very short-lived during storage. These seeds that are called intermediate seeds also need cryopreservation for their long-term conservation. Well, said so, when we are working with recalcitrant seeds, because other attributes that I have not mentioned in this presentation is that they tend to be very large seeds with a lot of water in the tissues. They are metabolically active, so they are very difficult to handle by the whole seed because of the size. We cannot cryopreserve the whole seed because it's a lot of tissue, is very large. So what we prefer when we are working with the seed, what we prefer to work with for cryopreservation is the embryonic axis. You know that the seed has an embryo. That embryo is formed out of the cotyledons, the shoot axis, the root axis, and usually when we remove the cotyledons, we have that tissue with both axes. That's what we call the embryonic axis, EAs, as I I make that acronym. And we prefer that tissue for two reasons. The first is because each embryonic axis corresponds to one seed. So when we are preserving genetic diversity, it's like preserving embryonic axis from individual seeds. It is equivalent to seed banking, because we can preserve an individual embryonic axis from an individual seed, so we can have a large genetic diversity with a lot of different embryonic axes from different seeds. All right, this is different to the clonal propagation and the clonal cryopreservation that was introduced yesterday. Here, what we want with the embryonic axis is to have a large genetic diversity by having a lot of different embryonic axes from different seeds. And the other positive thing about working with embryonic axis is that because in that embryonic axis you have the shoot and the root meristem within the same tissue, usually it's pretty straightforward to regenerate in vitro a full plant with relatively easy methods. You can germinate the embryonic axis, you can have a root and a shoot generally, and then you can acclimate that seedling and get a tree or a plant out of that test tube plants. Usually this is pretty straightforward, and you don't need to add any cocktail of plant growth regulators to induce shoot formation or root formation if you are preserving the shoot or the root tips. Well, so what are the common cryopreservation techniques that are available right now for embryonic axis of recalcitrant seeds? We published a few years ago in this cryopreservation and freeze drying protocols book from Springer, we publish a book chapter explaining the protocols we typically use, or we typically have tests for research. Right now, the editors, William and Harriet, they are working on a reedition of this book, and this chapter will be published again, but updated with more techniques that we want to include. But well, the basic protocol starts with the seed. We need to find the embryonic axis where the shoot and the root meristems are located, and once we have located them in whatever species is our target, they need to be excised from the rest of the tissues, whether it is the endosperm or the cotyledons, depending on the type of the seed. And usually during that excision process, we have to be very careful with the oxidation. We need some kind of antioxidant protections while we are excising and collecting those embryos. After we have a number of embryos collected, the typical cryopreservation method goes through a partial and rapid desiccation. Here you can see some images of a flash dryer that I will introduce later on. But usually you reach a target moisture content where the embryos, the water content is reduced in a way that we have very, very low amount of water content available for ice formation, but the water is still enough to avoid that the embryo dies. And once we have reached that amount of water content, that usually is determined empirically, we do a fast cooling in liquid nitrogen or in the slash of liquid nitrogen. Sometimes, depending on the species, we need an additional step of cryoprotection, where the embryos after excision they are cryoprotected in wherever it is the cryoprotective agent that works better for the species. It can be glycerol, sucrose, a combination of both, which is PBS3 or even PBS2 or whatever CPA is better for the species. And then usually going through the partial desiccation or directly into liquid nitrogen, we complete the protocol. Last year, Manuela Nagel, Bart Panis, and other colleagues, we put a review paper in an annual review of plant biology, where we commented on the cryopreservation of different tissues, including embryonic axis. And in this paper, we got this figure showing for all the species that have been researched the cryopreservation of the embryonic axis, what has been the success? We found out that about 66 species of recalcitrant seeds were studied for, well, were success in terms of cryopreservation, and in these 66 species, 40% were from temperate origin, mostly species from the genus Quercus, and 60% of them were from tropical or subtropical origin. What we found in the results of the cryopreservation is that usually with the recalcitrant seeds, we get a really low success. All right, we get only in between 0 and 60% of plant regeneration, with an average around 45% in this subtropical, and a little bit less in the temperate species, which is not very encouraging based in the research papers that have been published so far. And also maybe the reason because the implementation of the cryopreservation of embryonic axis is very low in terms of agricultural species, horticultural species, and also in terms of wild species, forestry species also including, where we found quite a lot of success in the cryopreservation of embryonic axis is in those seeds that I introduced before and are known as intermediate seeds, and that includes citrus, coffee, walnuts, and many other species. Here I present some of the results we have had in embryonic axis of Quercus and Aesculus species from temperate origin. We did a comparative study with in between 7 and 14 species, depending on the genus, and we got a lot of variation in our results. For example, in the case of Aesculus, we got really good success with Aesculus hippocastanum and Aesculus californica, with 70-80% of regeneration after liquid nitrogen, and other species with lower success, which were Aesculus assamica and Aesculus indica, with a success in between 40% or 5% in the case of Aesculus indica. We got also success in some Quercus species with Quercus pyronicus and Quercus rubra, showing 50% of regeneration post-cryo, and other species that are quite recalcitrant, not only to the desiccation but also the cryo, which are Quercus robur and Quercus suber, with really low rates of plant regeneration. This is quite common when you do comparative studies, and when you check these numbers that I was showing over here. You have a lot of variation depending on a species within a genus or within a family, or even between populations within a species. And that is one of the challenges we are concerned about the cryopreservation of embryonic axis, which is the first step, the step of the collection, the step of the plant quality. It is important to have seeds on its optimal quality during harvest, which is not always easy with recalcitrant seeds, especially those from wild origins. Also, we need embryos that are mature, and sometimes this maturity, this partial tolerance to desiccation or to cold, it is determined by ecological or phylogenetical characteristics. So there are some papers out there in the literature showing these issues in terms of the variation of maturity, or variation of tolerance to desiccation or freezing across recalcitrant seeds. For example, I have these two examples here, one in Aesculus hippocastanum and the other in Acer pseudoplatanus, both species with recalcitrant seeds. But the authors that you have in these works, mainly Matthew Dawes and colleagues, what they found is that the degree of maturation of the seeds, or the degree of desiccation tolerance, changes depending on the populations where those plants grow. And they found that these species were when growing southern in Europe, where you have warmer temperatures during summer and for longer times, those seeds tended to be more tolerant to desiccation. Also, they tended to have larger seeds with access with lower water content, and also access that tended to accumulate more solutes. Also, these characteristics suggest that those seeds growing in north environments, because the range of warmer temperature is shorter, they tend to develop seeds that are less developed in maturity, and that makes them more sensitive to drying, showing that these species growing in southern environments, because they have higher desiccation tolerance, they tend to be more prone to be cryopreserved. We also, with our results on Quercus species, we also try to find why some species were more prone to cryopreservation than others, as I showed you in the previous slide, where I show you that some species were successfully cryopreserved while others not. And one of the things we wanted to test was whether the environment was affecting to the response to cryo. So here you can see some of the climatological parameters that seems to modulate the response to survival after cryopreservation, and we have a positive relationship with the temperature of the warmer month, and a negative relationship with the annual rain, showing that the species in Quercus from drier habitats with warmer summers tended to survive cryo better than those from wet and fresh habitats. Some other studies, also in Quercus, have tried to find how the environmental parameters regulate the response to cryo. And in this case, Xia and colleagues, they found that the ability to avoid freezing in the species, so those species growing in really cold environments and dry environments in the high mountains, it seems to be a more important selection factor to increase desiccation tolerance of the seeds and the embryos, and hence to favor the response of cryopreservation. With Aesculus species, we also try to investigate this kind of variation in responses. We didn't find any environmental response. We didn't find any response depending on the size of the embryo and other physical attributes. But what we do find was some kind of relation in which one of the branches of the phylogenetical tree of this species, the Asian clade of the Aesculus, tended to be less tolerant to the cryopreservation protocols than those species that came from the European American clade of the genus. Well, apart from the challenges, depending on the variation of the species and the populations during collection, we found challenges in the cryopreservation of embryonic access in terms of the excision. When you excise the embryo, you are applying a physical damage, and also there is immediately a problem with oxidation. So we need to cope with those problems. You have in this photo down here, the black color in the embryo is how oxidative stress progress in the embryo as time advances from excision to 240 minutes after excision. This is in Castanea sativa. There are some ways, some methodological advances that have been investigated to cope with that problem of oxidative stress after excision. One that was developed in South Africa by Patricia Berjak and colleagues it is to when we are excising the embryo, it is to leave part of the cotyledon or the tissues around the area of excision, leave them paste to that explant. So we are making the damage in the cotyledon, not directly in the tissues nearby the axis, and that seems to postpone the oxidative stress inside of the meristems, and seems to favor the success of cryo in this type of tissues. Other ways to cope with oxidative stress, as you can imagine, is the use of antioxidants. There are different traditional antioxidants like ascorbic acid or citric acid that have been used to postpone oxidative stress during the collection of the explants, and also developed in South Africa by Patricia Berjak and colleagues. You have this cathodic water, which is a strongly reducing and high pH cathodic solution made out of an electrolysis of a solution made out of calcium chloride and magnesium chloride. These type of antioxidants, they help a lot, and they are needed in this step of excision, but also in other steps to reduce the oxidative stress, which is a high problem during the cryopreservation of embryonic access. Also, when we cannot cope or when we cannot work with the full embryonic access, because as I said, it's really recalcitrant. One of the examples is Quercus robur. I don't know if you remember the numbers I provided, but I was talking about 3% of regeneration success when you are working with the full embryonic access. Sometimes the problem is because during cryopreservation, one of the axes responds well to cryo, but not the other, and because the other is damaged, then you affect the growth of the full explant. So another approach that have been developed for recalcitrant seeds is to cryopreserve only the plumules, so only the shoot apex, the embryonic shoot apex of the embryo, and for example in Quercus robur, Quercus petraea, has been relatively successful, the cryopreservation of the embryonic access through a protocol including cryoprotection with sucrose and also fast cooling in liquid nitrogen. And in Poland, they are preserving this species in the Genebank, in the forestry Genebank, through the conservation of the plumules rather than the whole embryo. So this is another way to approach cryo of embryonic access if the full embryonic access cryopreservation is not very successful. More challenges that we can find is during the treatments, during the cryoprotection, because I have been saying that most of the protocols involve a desiccation, a partial desiccation process, sometimes with the use of cryoprotectants, but not always. And one of the reasons is because for what we have found that the use of PBS2 tend to be very toxic in the embryonic access, and tend to be very unsuccessful, the use of PBS2, which is the typical cryoprotectant used in many shoot tips and other tissues. So instead, other substances need to be used or need to be researched. And one of the things we have seen is that the use of cryoprotectants, as you can see here in this graph, allows higher regeneration rates compared with only drying, especially when the drying is slow. This is one of the examples that I can show you about the use of cryoprotectants. For example, in Amaryllis species, is a study also done by Patricia Berjak's group in South Africa, where we can see that most of the species they investigated, the regeneration, the viability after cryo was higher with cryoprotection, usually using glycerol only, rather than only by drying. The use of PBS2 in all these species was completely deleterious. I mean, it was deathly for most of the species. We also tested glycerol in these Quercus robur seeds, embryonic access, and we found that even though we could not have really great numbers, the proportion of regeneration could be increased significantly, both in the shoot and the root meristems of the axis. We also found that PPS2 was toxic for this species and for all the Quercus species we studied. To avoid these problems of toxicity of PBS2, there are other methodological or innovations, and one of them is the use of vacuum to infiltrate the cryoprotectants during the cryopreservation step. There is a paper that was put by Jayansi Nadarajan and Hugh Pritchard in 2014, and they show that compared with conventional vitrification using PBS2 and the embryonic access of Lauris nobilis, where you can see that you need in between 60 and 90 minutes of exposition of PBS2 in order to be effective. But you can see that there is a reduction in viability during the exposition to the cryoprotectant due to this toxicity. But what they found is that if you expose the access to the cryoprotectant inside a vacuum chamber, what is they call vacuum infiltration vitrification, you can get the full permeation of the cryoprotectants in less than 5 minutes, and the regrowth, the success of cryopreservation is very high. Compared with that of the conventional vitrification. So this is an innovation, a technological innovation that seems to work pretty well for some embryonic access of recalcitrant seeds, and is something that we should be testing also in those species for which only dehydration is not working very well. In terms of the desiccation step, which is the one mostly used, I have said that we need to reach a proportion of water contents in which we don't affect viability of the explants too much, but we reduce the water till a point where ice formation is not very problematic. In this review that I was mentioning you, in annual review of plant biology, we have this figure showing what is the water content range in which we have normal plant growth. And as you can see from the graph, there's a region over here in between 0.15 grams of water per grams of drying weight and 0.4, where most of the success in the cryopreservation of embryonic access is located. This yellow area over here is the area in which you cannot observe water freezing in differential scanning calorimetry, and this range over here is where you have freezable water, but below 0.4, the ice crystals that are formed are very small, and they are also, there is also a low number of ice crystals, and researchers believe that embryonic access can cope with that small formation of ice crystals during cryo. One of the things that we have seen in the literature also is that the faster you dry the explants, the better in terms of plant regeneration. So we tend to use this kind of flash drying techniques. Here I'm presenting some of the results we have in Aesculus and Quercus with water contents in between that range that I just show you. Really good success for this species, and this is the type of ultra fast drying devices that people tend to use in the literature. They call them flash dryings. Basically, there are two ways to construct these flash dryers. One is with a computer fan that on top you have the embryos inside some kind of container with a mesh, and then the air flows. You have silica gel on the bottom, so this dryer flows through the embryonic access, drying them pretty fast. Similarly, you can do flash dryers connecting a nitrogen gas through this kind of device with fish tank diffusers, and also some kind of container having two meshes, and also this kind of dry air of the nitrogen also dries the embryos very fast. The next challenge we can find during cryopreservation of embryonic axis is the cooling. We need to avoid a large ice crystal formation and growth and induced vitrification. And one of the main things that have been investigated is the use of different cooling rates. Generally, the fastest is the cooling rate, the better success you get in terms of the regeneration of the embryonic axis. Although there are some low cooling rates that tend to work pretty well for some species, depending on the protocols. Some of the ways we have to increase the cooling rate, it is by using some kind of slash of liquid nitrogen. You solidify the liquid nitrogen inside a vacuum chamber, and then when you release the vacuum, you get this kind of a slashy liquid nitrogen that is minus 210 degrees Celsius compared with minus 196 of liquid nitrogen, and you can get fastest cooling rates if you, for example, put your axes inside of aluminum foil or you inject them directly in that kind of a slash. There are different techniques that you can find in the literature. And finally, during the storage, we need to avoid changes in temperature that may induce devitrification and formation and growth of ice. One of the questions that sometimes remain open is whether the cryopreserved access how long can they survive in these conditions. And for example, we did an evaluation a few years ago comparing a long-term storage collection of Aesculus juglans trees, the embryonic axis, as you can see here. And after 2 decades, we rescue them and we compare viability with other data that we have from 10 years storage and from freshly harvested and after drying. And as you can see, for most species we tested, except for this Aesculus glabra, the regeneration of the plant was the same than initially. And also we got healthy plants out of those embryonic axes. In Juglans nigra, we were not able to do it in Aesculus hippocastandum, but I think it was more a problem of acclimatation rather than the quality of the plants that we got. In Aesculus glabra, although we didn't get a lot of survival, maybe because of the tube that we tested, what we got was a lot of embryonic tissue production out of the embryonic axis cryopreserve. Again, with the warming, we have to be careful. It needs to be performed fast. We need to protect against oxidation after warming. And for oxidation, I already mentioned the use of antioxidants, including this cathodic water. And for fast warming, not in embryonic axis, but in other plant tissues, it have been tested the use of gold nanoparticles and other techniques in order to improve the warming process and speed up the warming process. Generally, you used to do the warming in a water bath at 40 degrees or immersing the embryos, or even immersing or immersing the cryotube, or immersing the embryos directly into a solution that is warm to 40 degrees Celsius. Before I finish my presentation, because I think I'm close to my 30 minutes time, I wanted to introduce this slide indicating that sometimes when we have a species or genera that produce recalcitrant seeds, we have another option to preserve this species, and in this case is the use of the pollen, at least to preserve the male gametes or the male genotypes through the pollen. Because, for example, through different research we have done in the United Kingdom, in the Garfield Weston Tree Seed Bank project, and also in this Global Oak Pollen Bank funded by the Oak Conservation and Research Fund of the International Oak Society, we have found that most of the Quercus and Aesculus species if not all, produce pollen that is orthodox. We did this study testing 20 Quercus species from all around the world and 7 Aesculus species, and we found that they tolerated drying up to 11% relative humidity. When they were dry, they tolerated storage below 0, including liquid nitrogen, with maximum longevities achieved in between 11-30% relative humidity, which is what orthodox seeds tolerate. The only problem is that pollen is pretty short-lived, so we need to be processed very fast, and we recommend liquid nitrogen storage for long-term preservation. With those parameters, we are working on the formation of a global oak pollen bank using different institutions across the world, and that is ongoing. In my presentation, I needed to talk also about the cryopreservation of orthodoxies that are short-lived, and I also mentioned the intermediate seeds, for which you don't necessarily need to excise the embryo, and you can preserve the whole seed. There are protocols that are available in the literature in this book, Cryopreservation and Freeze-Drying Protocols, that I mentioned before. There are 2 chapters, one dedicated to seeds and embryonic access, and the other to pollen and furnace spores. And basically, the protocol is the same for this species. When you have orthodox seeds and or orthodox pollen, after collecting your explants, these pollen or these orthodox short-lived seeds are dried to relative humidities in between 30 and 40%. This is slightly higher than the relative humidities used for orthodox species in conventional banking at -20, and that is made on purpose to avoid over drying for tissues that are banked at cryo temperatures, but they are equilibrated at room temperatures. All right. So to account for that, we equilibrate at higher relative humidities. Then the seeds or the pollen is stored in the vapor phase of liquid nitrogen, generally because the cooling is a little bit slower than immersing directly in liquid nitrogen. And then you can do the storage in liquid nitrogen, of course, but for those that have not liquid nitrogen facilities, a -80 mechanical freezer can also be used. That's for these orthodox seeds and pollen that are short-lived. Then the warming can be done at ambient temperature or in a water bath, and then you do the standard seed germination. When you have intermediate seeds, the problem with the intermediate seeds, whether they are citrus seeds, coffee seeds, jugulant seeds, is that they don't tolerate the full drying of an orthodox material. And for example, while jugulants tend to be dry in between at around 50% relative humidity before immersing in liquid nitrogen. For other species like coffee and citrus, the drying is done in between 50 and 85% relative humidity, depending on the species, and then you do the fast immersion in liquid nitrogen. For some coffee species, you need to do a slow cooling up to -50. If you have a programmable freezer or a Mr. Frosty, and then the freezing in liquid nitrogen. Usually these tissues, these intermediate seeds, they are warm also at high temperatures in a water bath. As I said, you can find the protocols pretty well detailed in this guidance, and I'm happy to share with you these chapters if you don't have access to the book. And when with all this information said, I would like to thank all of you for attending the webinar. Thank you, the organizers, for inviting me to do this webinar, and all the colleagues and students that have been working on the development of these protocols that I have been explaining to you today. Thank you very much.
Wow, thank you very much. Thank you very much, Daniel, for this wonderful presentation. So without further delay, I will give the floor now to Rainer and Bart for the follow-up presentation.
Okay. Thank you, Dani, for this very nice presentation, by the way. In fact, Rainer and we thought building up further a bit on what we discussed yesterday. So we gave a lot of theoretical content on cryopreservation, what the methods are to be used. Also, Danny gave a very nice background on that. And we thought it maybe not bad just to give an overview of our cryopreservation activities practically. I think that Rainer already highlighted that yesterday, but I will highlight a bit the cryopreservation program of the banana gene bank. So I'll share my presentation or try to share my presentation. One moment. Okay. Oh, that is not working. Hey, why? So sorry. Oops.
While we are waiting for you, don't worry, but I just want to inform the participant that the Q&A session will be at the end of all presentations, please. So, thank you.
Okay, I think I almost have it. Yes, you see my presentation?
Yes, yes, we can see it. Just
Okay.
now put in the presentation mode, please.
Okay, I shared the wrong screen. One moment. Yeah, stop. I have to do this then. You see it now?
Yes, we can see the importance of banana planting.
Okay, but I first.
Yes.
Okay, so I will give a bit of information on the cryopreservation program at the Alliance. So it's already highlighted yesterday, the importance of bananas and plantains. It's not only a fruit eaten as dessert as we do in the West, but it's also very important starch source in Central East Africa, in Latin America, in Asia. It is the largest food crop in the world, almost double the production compared to number 2, which is apple, and it is international trade over is quite high. Method of conservation. Yes, so we have in situ conservation.
Without stopping you, Bart, please, can you put it on the presentation mode?
Oh, okay. Sorry. Oops. I thought so. You don't see the presentation mode.
Okay. No, not on my side. I don't know if somebody else can see it. Okay, then go ahead. Yeah, I thought it was. Go ahead.
It's okay. You see the slides?
Yeah, I can see your screen now, not the slide.
Okay, like this?
Yeah, this one is good now. Yeah,
okay.
Is the one. Yeah, go ahead, please.
So you see methods of conservation on top. Yeah?
Yes, exactly.
Okay,
yeah.
Okay. So we have in situ versus ex situ. So in case of edible bananas, seed collections are not possible because bananas do not form seeds. So field collections are good alternatives. So you have field collection, for example, in Costa Rica, Corbana field collections. Then you have in vitro collections, as we have here the big banana collection as shoot tips grown in a cold room at 15 degrees Celsius. And then we have cryopreservation banks. Okay, so what is the situation of our banana collection? So it was established in 1985. It's hosted by the University of Leuven here in Belgium. So we have an in vitro active collection of 1,716 accessions. So all the baskets that you see is one basket is one accession. 1,328 of these accessions are for the moment stored in liquid nitrogen in our cryotanks. And out of these 1,328 accessions, 1,181 accessions are stored as a black box safety backup in Montpellier in France. So why do we do that? Suppose something goes wrong with the in vitro collection and with the cryo collection, because they are situated in the same building, and we can always re-establish our collection from the material which is stored in the south of France. So that is the ultimate security. We have, besides that, we also have a lyophilized leaf tissue collection. In fact, these are leaves from bananas which are freeze-dried and which can be distributed simply in an envelope. But of course, this is not living tissue, this is dead tissue, but this is good materials for people who are doing molecular biology. Where is the material coming from? Of course.
But we lost your
Yes?
presentation. We lost your presentation. We can't
Oops.
see you.
Again, sorry. Okay, I'm making a mess out of it. Okay. Now? You see now
Not
presentation?
yet. Not yet, just the screen, the blue screen.
Okay, I will try again.
Now it's perfect. It's full screen.
Okay.
Yeah.
Okay. So the origin of genetic resources. So where is the material coming from? Most of the material is coming from Southeast Asia, but they also have some secondary center of diversity, namely East Africa and Central Africa. And then, of course, you had also institutes that already had a gene bank and that were supplying us with in vitro materials. Let me go to the next slide. Okay, see my next slide? Yes. So our protocol is based on the droplet vitrification of rooted shoots. So we start with a in vitro plant, a banana in vitro plant, a very strong plant. And from this in vitro plant, we excise the small apical meristem. So what in fact we need to do under the binocular, under the microscope, we need to remove leaf by leaf with a scalpel until we end up with this meristem. So the diameter of that meristem is about 1 millimeter. So you can imagine it's quite labor intensive to excise all these meristems. Then the meristems are treated with the cryoprotective solutions, in our case, loading solution and the vitrification solution, PVS2, placed on a piece of aluminium foil and then plunged rapidly in liquid nitrogen to have ultra fast freezing and that way to prevent lethal ice crystals. So this is how it looks like, the protocol. So the freezing. Then when after storage, the material, so the pieces of aluminium foil are taken out from the liquid nitrogen and rinsed in recovery solution, so at 25 degrees. So this is fast recovering, like I also mentioned yesterday. In 2 seconds time, you go from the temperature of liquid nitrogen to room temperature, and then you regenerate the plants. So the protocol that we use for banana can be found back in these technical guidelines. It's available online. So it's the protocol is described in detail there. So then something on some practical issues. So it was already discussed yesterday. Yeah. So how many accessions can one technician cryopreserve per year? In case of banana, this is 45 accessions per year. Does not seem very much, but we have certain standards. So we consider an accession as safely cryopreserved if we did 3 independent successful repetitions. So one repetition is consisting of 48 meristems. So 48 meristems means that for 48 plants, you need to excise these very tiny meristems. 3 times 10 of these 48 are going in the cryo tank for eternity maybe, and 3 times 6 are recovered after 1 hour of liquid nitrogen exposure. And it is with these this material that this we consider as one repetition. And we need to do this 3 times, and it has to be 3 times successful to consider the accession as safely preserved. What do we consider as a successful repetition is that we are 95% sure that at least one plant can be regenerated from the repetition. So we have some statistical calculations there where we will fill in some numbers and where we get some probability numbers. So 3 times we need to more than 95% sure. The 3 repetitions are stored in 3 different cryotanks. 2 of these tanks are located here in Leuven, and 1 is the safety backup in Montpellier. Just to give you an idea how much time it takes to cryopreserve a collection. So here you see we started in 2003. Here you see the increase of number of accessions cryopreserved, and now we are here at 2023-24. Yeah, but you see that sometimes it goes fast and sometimes it goes slower, and this is of course to do with the number of colleagues that can do the cryopreservation. So if here at a certain moment had 4 people doing the cryopreservation, here only 1 person, and here again 2 persons. So you see, it's all depending on labor. And of course, labor means funding, and funding means that you have to apply a lot of projects to different funding organizations. Here you see some examples. The cryo protocol that we developed for banana droplet vitrification is quite robust, so it is applied to many other plant species. In total, we can cryopreserve now about 40-50 plant species using the droplet vitrification protocol. Also, the sweet potato collection in SIP of Reiner is also cryopreserved using that methodology. We also not only working with shoot tips, but we also now working with seeds, which we also cryopreserve. So we are doing some tests now, comparing different storage methodologies of the seeds. Room temperature 5 degrees to -20, -196 degrees, and you see that the banana seeds seem to be quite orthodox, so which is good news for us. The only problem is that you need to have mature seed. And also we are working now with pollen storage, which can be also quite important, as Danny already mentioned before. We do a lot of training on cryopreservation, also like SIP is doing. The equipment that we have here, but later we will come back to that. So we have big cryo tanks, somewhat bigger than others. So these are the 2 huge ones, which can contain about 36,000 cryotubes. The others can contain about 5,000 cryotubes, 2 milliliter cryotubes. We have dry shippers, control, filling system, but I will come to that back later. So this is a bit the overview of our cryo activities in Belgium. Okay. Rainer, do you have something to add there from the side of SIP maybe?
Well, I think you said everything. Of course, as you mentioned, the crops are very different. As it's easier to excise a potato shoot tip than a banana shoot tip. Therefore, a person can cryopreserve more accessions per year. And this changed a little bit the number of vials you store, but this is just a consequence of the different crops.
Yeah. Okay, then maybe I can go to the few slides that Rainer and me gathered on practical considerations, what you need, for example, if you want to start with a cryobank. I will try to open my presentation. Okay, so you see my slide this time? Marie-Noël?
Yes, you can see it,
Okay,
yeah.
good. Okay, so some practical consideration and challenges of a cryobank. So we in fact considered different things which seem to be essential if you want to store it with a cryobank. Okay. Okay. So the first essential thing is liquid nitrogen, of course. I mean, without liquid nitrogen, you cannot start cryopreservation because you need this ultra low temperature to store material for the ultra long time. So there are two possibilities. How can you have in Leuven, we buy it from a company, Air Liquide in our case, and this is very cheap. I mean, just to give you an example, I think we pay around 0.05 euro per liter, which is almost less than water. It's extremely cheap. This is why we also do not have a liquid nitrogen generator. But in SIP, on the other hand, maybe Rainer, you can tell something about the generator, liquid nitrogen generator. You have them, eh?
Yeah, the situation here is different. 1 liter costs about $2 now.
Oh.
Also, therefore, yeah, we have invested in a liquid nitrogen generator to reduce costs. You can also buy it there, but I think you said 0.05, so
Yeah,
that's 40 times more expensive in Peru than in Belgium.
yeah. And okay, here you could also say, look, but you are really depending on one source. For the moment, that is true, but it's the same supplier that also provide liquid nitrogen for the university hospital and a big hospital. So the supply should be secured. So we are quite sure that we will have always liquid nitrogen at a good price in our case. Okay, so the second thing is, of course, equipment. And equipment, if you want to do cryopreservation, this can be all kinds of things. So liquid nitrogen is a dangerous gas. It is, you can smell it, you can't see it. And it replaces oxygen. So this means that when there is a leak in a tank in a closed room, and the room begins to fill with the nitrogen, you can suffocate. So this is why in a room where the liquid nitrogen tanks are situated, you need to have oxygen meters. It's for security. What else? You have, of course, the tanks. I will go together with Rainer to that later. You have, of course, if you are working with in vitro cultures, as is the case in most cases, you need laminar flow benches to work in sterile conditions. You need culture rooms. You need an autoclave. And also in the cryo room, a direct piping, for example, between your big nitrogen tank outside and your tanks inside is also something helpful. One other equipment we see here is a dry shipper. A dry shipper is a equipment that can be used to transport material at ultra low temperature. Because you cannot use a normal tank for that because liquid nitrogen you can just not transport in any vehicle. It's forbidden. But when the liquid nitrogen is immobilized in a kind of sponge, which is in such dry shippers, then you can transport the materials. So we use these dry shippers, for example, to transport our cryopreserved meristems from Leuven to Montpellier in France. So Rainer, you also use dry shippers, yes?
Right. We have our safety copy of our cryo collection in Huancayo in the highlands, and yeah, we use cryo shippers also. And yeah, for the future, also for the international safety copy, you can ship them also with air, also with the airplane. Yeah, they're very useful. Yeah.
Yeah, so there are special companies, in fact, that are specialized in doing such transports. World Courier is one of the examples. So, what is the size of your dry shippers, Rainer? How many tubes can it contain?
Oh, it can contain 2,000 vials.
Okay. These are huge ones then. These are quite new. Yeah. Okay. Okay, so then you have, of course, a very important thing, the tank where you store the cryotubes. Maybe, Rainer, you can explain a bit?
Okay, so from the left to the right, it's getting smaller, as you can see. No? Large cryobanks as Bioversity and SIP use for their final storage tanks like this on the left side. I think from Bioversity is even larger than this one. One of these tanks, as a 20,000 vials, costs about $40,000. And if you store 10 vials per accession, you can store 2,000 accession in one of these tanks. Then the next one, they are very useful. I think you also have one of these, no? The MBE 6000. I've seen it in a photo.
Correct.
As this, we like them a lot. Now it's space for 6,000 vials, $20,000, 600 accessions. So this is a good option to scale up a little bit, no? But then the next three ones, we would consider that this is let's say the first step, the basic tank equipment. You can start a cryobank, no? And as you can see, it's not so expensive. No, you need one tank for final storage. In this case, this tank could contain 2,000 vials, 200 accessions, $10,000. But it's useful also to have to a tank. The three tanks on the left side are all cryobox based. So they have a rack, a stainless steel rack, and this rack contains boxes, and within the boxes there are the cryo vials, generally 100 vials per cryobox. But then you need also something more practical in the lab for lab storage. It's cheaper, $6,000, and then you store the vials on aluminum canes within small vessels. On the right side, you can see one of these vessels right to the tank, and there you place in the aluminum canes, and these are then submerged in the tank. And then you, of course, you have to fill up your freezing container when you work in the flow hood, and this is a 4-liter liquid nitrogen tank. And on the right side, you have a cryo shipper. This is not so important when you start, but if you imagine a flow chamber costs about, I would say, between $8,000 to $10,000, at least in Peru. So $20,000 to start doing cryo is not so expensive.
Yeah. Okay. Okay, thanks. Then a very important point is skilled staff. So I've shown you how difficult it is to excise these meristems. So you need staff that is skilled, that has some experience to really reach enough materials that can be cryopreserved. So if you have good staff, value them, try to keep them, because yeah, it can save you a lot of time. An efficient cryopreservation protocol, of course, that's also handy. What is an efficient cryopreservation protocol? Of course, it is a protocol that gives high plant regeneration after cryopreservation, and with the emphasis on plant regeneration, because survival is not enough. In the past, there were many reports where after cryopreservation, only survival was reported. And later on, that survival could not lead to plant regeneration. So very important is, if you do cryopreservation, make sure that you can regenerate a rooted plant of the materials, because you can sometimes have a small shoot surviving, but it will not grow any further. And this is really also depending on the plant species that you work with. Then also important is that the protocol you establish is applicable to all or most accessions from a specific crop. So it's possible that you have some accessions that are better responding than other responding, even from the same species. So make sure that you make a protocol that is applicable to all of them. And then, of course, a good efficient cryopreservation protocol is also a protocol where you can manipulate as many propagules as possible. In case of banana, for example, the excision of the meristems is a bottleneck. One can only excise, let's say, 15 meristems per hour. In case of potato, this is 4 or 5 times as high, I think. So number of propagules is very important. A good planning. This is something, and I think that Rainer will confirm that, is essential. We have seen it before that we train scientists. They are very skilled. They know how to perform a good cryopreservation protocol. But then when returning, they do not reach, or it's difficult to have this theory translated into a cryobank. And this is often to do with planning, because you have to take into account that when you do cryopreservation, you have to see that culture media are prepared in time, that you have enough plants multiplied in time. Only you start sometimes with 2 plants, you need to multiply them up to 100 plants. You need excision of meristems. You need a cryo protocol, which takes a half a day. You need to do observation, data management, filing, control of liquid nitrogen levels. So you need to plan all these things. Otherwise, it becomes very difficult. And this is something, where I think we visited the cryobank in SIP end of last year, and this is really an example of good planning. And then, yes, finally, long-term funding. Cryopreservation, we are not doing this for the next 2-3 years. We are doing this for next generations. So you have to consider that you need some funding for the long term also. Because once, for example, the liquid nitrogen is finished in the cryo tank, all the material dies. So you need to be sure at least that you have funds to maintain the material in liquid nitrogen. The funding also, if you want to cryopreserve a big collection, if you can only cryopreserve 40 accessions per person per year. If you have a collection of 1,000 accessions, yes, you need 25 person years to cryopreserve this. Yes, I think these are the main points. Maybe Reiner, you have something to add there?
No, but I think it's all said.
Okay, so with this, we wanted to give you a kind of overview of what you need to buy or need to purchase before you start with cryopreservation, some practical challenges. Yes, I think that's it. Thank you. Any questions? Marie-Noël? Marie-Noël seems to...
She left. There's a question in the chat about, it says the names of some equipment suppliers. Well,
you have MVE, you have Air Liquide. I don't know what are the brands that you are using, Reiner?
we all our tanks are from MVE, but previously we used Taylor Wharton, I think.
Yeah. But you can find them on the internet. But most of the equipment is I think quite good. Danny, you use specific equipment?
We use Nippon gases or Air Liquide here in Spain mainly, and the cost of liquid nitrogen is about €1 per liter, or a little bit less depending on the amount.
Yeah. Okay. Wait a minute.
Then another question is, what about the challenge of power supply in some regions? And Elizabeth has her hand up. I don't know how we can. I can't. I think only Maria can unmute her. Or Elizabeth, can you talk? Can you unmute yourself?
I think we can by clicking right.
Yes,
Yes.
I think.
Okay.
Hello,
Please.
good morning, all. I think Marie is frozen, but she's working in the background. I just want to say thank you so much. I mean, these have been fantastic, the amount of information that you packed into these presentations. I have a couple of questions, especially on the pollen preservation. When you preserve pollen, you're only preserving half the genome and you are not preserving the mitochondrial genome. So how are we going to recuperate the ploidy level back, and how are we recuperating the mitochondrial genome? Why are we not preserving ovules instead, for example? So that's one of the questions. And the other one I have is, I know it's impossible to go through all of the species that are, going through the cryopreservation process at this time, but are you at all considering medicinal plants?
Sorry, I was... Okay, thank you for taking over. My connection was
frozen.
Yeah, please, yes. Thank you so much for taking over. So just go ahead, please.
Yes, thank you, Marie. So yeah, my question was on both the genetic loss in storing pollen and also medicinal plants. Are we also looking at medicinal plants? That's it. Thank you.
I can respond to both questions if you don't mind, Bart.
No,
so
perfect.
in terms of the pollen, we are proposing the creation of pollen banks for these recalcitrant species, for which nowadays there is no collection there are no collections of any way. So if you go into conservation of Quercus, which is a large genus of temperate trees forming forests, important in the wood industry, in the forest industry, many industries. So there's no long-term backup collection for oak species and the genetic diversity. There is a cryobank in the north of Spain preserving a few genotypes of high productivity in a couple of species, and that's all. So obviously we need mothers, we need ovules, as you said. We can do that relatively well using embryogenic tissues, but that is more a clonal approach. Some people is working on cryopreservation of shoot tips also, but it's difficult because of the oxidative stress, as I said. But it seems that with embryogenic tissues, cryopreservation is possible for oaks in this case. So you could create a ovule or a mother's bank using specific genotypes or a small number of genotypes using these clonal approaches. But if you want to preserve biodiversity, a large number of different types of genotypes, is where the pollen bank is very useful, because you can, even though you are preserving only the males, you are not preserving the mitochondria, you're preserving a lot of the nuclear genetic diversity that later on you can reinsert in those mothers preserved in another way, or even though in arboretums, in living collections, by doing crosses. And that's really useful for getting some of those genotypes preserved. For example, we got experience with Castanea dentata, the American chestnut, which is mostly extinct in the wild. And there is a foundation, the American Chestnut Foundation, doing preservation of some trees, but they really need genotypes of those trees. And we got pollen cryopreserved for 20-something years in the bank of genotypes that were extinct in the wild, and we were able to put back those, at least the nuclear genes, that we were able to put them back into seeds, into a progeny of trees that got at least that part of the genetic diversity. So it's a way to improve the genetic diversity of other collections, either on living collections or clonal collections like shoot tips, embryogenic tissues, and so on. And what was the other question?
Medicinal plants.
Sorry?
Medicinal.
Oh, medicinal plants. Yes, there is a large interest in conserving medicinal plants through seeds, either if they are recalcitrant intermediate. And I think the people who is doing that in a better way are the Indian ICAR. ICAR, they have a very good cryobank, and I know they have a few reviews out there. They just published a review on the Indian cryobank in cryobiology in the journal. In a special issue that I was editing, and they review all the species they have in cryo in terms of seeds and seed embryos, and they work a lot with medicinal plants from that region.
Okay, thank you. Just to go back to the pollen. So, are you collecting the pollen before anthesis? At what stage is that pollen collected?
Yeah, that's one of the tricky issues. You need to understand the species. It's easier with wine-pollinated species than with animal-pollinated species. For example, with the oaks, it's really easy to collect the catkins before anthesis. Right before anthesis. So, for example, we did a study on when the flower is mature enough to have high quality pollen in terms of germination, in vitro germination, and overnight you leave the catkins on the bench, and they release all the pollen, and you collect it very easily. But you need to check species by species. We collect before anthesis, and we store genotype by genotype in the bank to keep the different genotypes individually. And also we track the trees with GPS, so we know which trees from the wild we have in the bank.
Okay, so you're saying it works better with wind pollinated rather than insect pollinated or vector pollinated?
It's easier to collect the pollen, but it works equally because Aesculus is an insect pollinated species, and we also have been successful with that genus. The only thing is that in terms of getting the pollen, sometimes what you need to collect are the anthers before they open, and you dry them and you preserve them along with the pollen, the anthers, and then you do the pollination in another way. But with the wind pollinated species is really the collection step itself, but the preservation is the same for both.
It's really depending from one species to another. I mean, for example, banana pollen, you kill them most of the time by drying, because you need to dry before cryopreservation.
Yeah.
This is really depending. Also, you need most of the time to have mature pollen. Yeah, so you need, it's case by case. You need to investigate.
Yes, I mean, what the techniques I was showing was for orthodox pollen. As Bart said, banana and many other monocots, especially from the Poaceae family. So wheat, for example, a main crop, the pollen is recalcitrant, is highly recalcitrant. It's very difficult to preserve indeed, because it's very short-lived and very desiccation sensitive. Those species you need to do research on much more. I mean, what we are proposing is an easy and straightforward, a low-tech way of conserving recalcitrant seeds that have orthodox pollen, at least the male gametophytes.
Okay, colleagues, please. Sorry, I don't mean to dominate the conversation, but the pollen is very attractive in terms of, especially in the Caribbean, where we are looking at coconuts. And lethal yellowing is one of the things we don't want to move around. So, you know, being able to move just the pollen rather than the plant or tissue is very important. So I can pick that up with you at a different time.
Yeah, perfect.
Thank you very much.
And also Gail Volk in the USDA has been working a lot on pollen for crops. So she also have good guidance and information out there. I think they are preparing some kind of paper about their experience. So you will see that in the literature soon.
Yeah, I'd like to get her contact. Thank you very much.
Thank you. I can see another question on the role of triacylglycerols.
There is one, Hamad, who raised his hand. I don't know if you are already raised the question before we go to the chat.
No.
Please go ahead.
Thank you, Mary. I'm waiting. I was waiting, actually. I am in the queue. Okay. Yet another day with great presentations. Thank you so much for wonderful and knowledgeable presentations, Daniel and Bart. So it's a less question, but a point of discussion, a point to ponder, actually. That of course we are considering the efficiency as a major bottleneck. Today, Daniel presented a very good presentation, and he showed a very high percentage or very good efficiency, actually, with the species he is dealing. My point is, okay, so can we utilize the gene editing tool, CRISPR, or any other variant, the genetic engineering tool, to improve the efficiency of the crops or the genetic material that we are going to preserve using cryopreservation technology? Related to this point, what do you think? My question is actually, okay, yes, we can use Wushel or Baby Boom or any other genes or the stress sensitive genes. We can target them and we can improve the efficiency, regeneration efficiency. But what do you think about the regulatory aspect if we are using the genetic engineering tools such as gene editing? What do you think, how the policy or how the ethical consideration, whether it will be allowed, particularly with respect to Europe, whether at the moment they're considering new genomics technology and they're discussing the policy on it. So what do you think in future? It is, of course, a forward-looking question, and I would be happy to listen from your perspective. That what do you think? How the policies or the regulatory aspects can move forward regarding if we are going to improve the genetic efficiency or regeneration efficiency of the species or the genetic material that we are going to preserve using cryopreservation. And yeah, that's it. If I could
okay, maybe I can comment a bit on that. I'm personally not a great fan of manipulating material to be stored to conserve genetic resources, because CRISPR-Cas and or other genetic engineering methodologies are not so easy. I mean, to have one good edited plant, it takes you a lot of time. If you have a collection of 1,000 different accessions, for example, and you have to CRISPR them all independently, because and also conserving genetic diversity is conserving mainly the natural diversity. We should concentrate on that. So this is why in theory it could be possible, but it would take a lot of time even to CRISPR one individual, and you have to do this for 1,000 individuals. I don't see it. Of course, it's something different when you want to manipulate material to have a better yield or better disease resistance. That's another thing. But to increase the conservation, I'm not really a fan of that. I don't know, Daniel, what do you think?
In terms of research, it would be great to manipulate some genes like LIA proteins and things like that to check whether it is possible to increase the desiccation tolerance of some plants. That's something that is on discussion. We have tried to do it, but not through gene editing techniques. But as said, in terms of research, it would be great to see whether you can manipulate a seed to increase the contents of sugars, LIA proteins, and many other substances that are known to favor the preservation. But in terms of conservation of accessions, I wonder whether that's ethical in terms of those accessions that you want to cryopreserve, those unique genotypes, if you manipulate them all. That's something that escapes to my knowledge. And
okay, so thank you very much. It means no consideration about the regulatory aspects, particularly with respect to the conservation of using the genetic engineering techniques. This is what
you mean in terms of the research? I'm sorry.
Please. Yeah, we can conserve.
Yes, my question was a bit more research based on the side of research. That's correct.
But in terms of the research, I think it's a good approach that could be considered. As I said, there are a lot of groups. If you go to the cryo meetings or the desiccation tolerance meetings or the seed meetings, you always see a lot of research in terms of the genetics and the proteomics, the metabolomics of desiccation tolerance, and how one seed becomes tolerant or intolerant to desiccation, so you can make it orthodox or not. And there are a set of genes that are involved in terms of the tolerance to desiccation, in terms of the longevity also, and people is working with mutants in Arabidopsis, in Medicago, in many species, to try to understand those fundamental aspects of those parameters, the desiccation tolerance and the longevity, which are the main ones. And there are different sugars that are involved, different alcohols, there are different proteins like LIA proteins, heat shock proteins. Whether you can manipulate a recalcitrant seed to convert it in an orthodox seed by bioengineering, I mean, that would be great. I encourage you to work on those and on that direction, because it's something that it would be good to see if that's possible to be done. You know what I mean? So yeah, that can be possible. It's just to find a way to do it, which escapes a little bit to my knowledge and expertise.
Thank you, Daniel.
Perfect.
Please, only one question, please. I don't want only one question per person, because we are far beyond schedule, and then we have to hurry up. We still have a lot of issue to raise in the chat. Thank you, Daniel. Thank you, Ahmad. So now we have Amara, who is asking about the name of the supplier. So please, maybe when you share your presentation, you can add the name of the supplier so that they can have access to this information. So we have our Monsieur Charles, who is asking, what about the challenge of power supply in some region? Any possibility of cryopreserving crops without leaking nitrogen? Who want to?
Okay, I can. Power supply. I mean, the storage itself takes place in dewars, in thermos flasks. So we do not need power to preserve. The only thing what you need to do is regularly fill up the liquid nitrogen level. But of course, if you are relying on a liquid nitrogen generator, which needs power, then of course you can come into troubles. If you have 4 weeks no power, then when you finish your liquid nitrogen, then you are in serious problems. Of course, power is also needed if you want to do cryo, if you want to work on the laminar flow benches, you want to cool the culture rooms. But for the storage itself, you do not necessarily need a power supply. It's not like a fridge. Did that answer the question?
Charles, are you satisfied? You can, I don't know where it is because I have to enable the microphone. Anyway, you can write in the chat if it's okay. Then the follow-up question is from Yalenis. Dr. is asking to Daniel, I think, could you comment about the chemical, the role of
the lipids.
triacid glycerol in the storage quality of recalcitrant and intermediate seed during the decrease of temperature for conservation?
Yeah, that's a pretty important issue, especially with the intermediate seeds. One of the syndromes that define intermediate seeds, indeed, it is the freezing sensitivity of the seeds, which usually is related to the crystallization of the triacylglycerols. Okay. So there are seeds with a specific composition of triacylglycerols that during storage at -20, those triacylglycerols crystallize, and for whatever reason, viability is affected immediately or over time. So there is an issue there that is under the research. There are some papers out. I can recommend you some lectures. Crane et al., for example, in Cofia. There are works on citrus, on coffee, also Dussert and colleagues. So yes, there is an important role of triacylglycerols in the longevity and the storage quality, particularly of the intermediate seeds. For the recalcitrant seeds, there is not a lot of research linking those issues, but also because usually you go to very, very low temperatures where the crystallization of those triacylglycerols is not the main issue. The main issue comes with those triacylglycerols that crystallize at the banking temperatures at -20 in the intermediate seeds.
Okay, thank you. Please, if I can come back on the question of Charles, we still have the second one. What about the cryopreservation without liquid nitrogen? Is it possible to cryopreserve without liquid nitrogen?
Well,
technically, yes.
Please, Daniel.
I mean, you can do some storage at -80, indeed. Some materials that can be dried, you can lower the temperature to -80 without problems. There is a paper in this cryopreservation issue that I was mentioning that is out on people cryopreserving embryogenic tissues in PBS3, if I don't know, in DMSO at -80 for the long term. Pinus embryogenic tissues cryopreserved up to 6 years at -80. It's a paper that came out recently from a group of the north of Spain. And then there are mechanically powered freezers that reach liquid nitrogen temperatures, but those tend to be expensive and difficult to handle. So possibilities, yes. It is a matter of how expensive or tricky is the technology, or if your tissues allowed -80.
Yeah, it's also a question how far you can dry the tissue. I mean, if you have very hydrated tissue, yeah, the water can always crystallize, even at ultra low temperatures, at -80 even. So it's depending on how far you can dry, and then maybe you can have higher storage temperatures. But in case of let's say PVS2 storage, I would always suggest that you go below -130, because this is the cold crystallization, the crystallization temperature of vitrified solution. Yeah. Sorry, the liquidifying temperature of solid PVS2.
Then, thank you, Bart and Daniel. Then we have a question from Ibidun, who is saying that, can we use a solar panel as a source of energy for the cryobank? Will the temperature fluctuations affect the genetic integrity of the cryopreserved material?
Maybe I can jump in on this. If you want to create energy with a solar panel for a liquid nitrogen generator, then you will need many solar panels because they consume a lot of energy. To maintain the cryotank itself has a monitoring system, a liquid monitoring system, also the modern tanks. This requires only very few energy. So for this, a solar panel, small solar panel might work. But for a generator, I don't think also you would need a lot of space. And as we mentioned in our presentations, best thing when you are there and not sure with the liquid nitrogen, to have two sources. Maybe think about a small generator, but also ensure a liquid nitrogen supply, external supplier. So when one of the option fails, you have a plan B to go for.
Yeah, one must remember that when samples are cryopreserved and stored in liquid nitrogen, the liquid nitrogen is the coolant. You don't need energy to preserve the temperature. It is the liquid nitrogen that preserves the temperature. You need to maintain the level of liquid nitrogen. So in theory, as far as your liquid nitrogen is within the recommended levels, you don't have temperature fluctuations. You don't need energy to preserve that tank full of liquid nitrogen. You need energy to produce liquid nitrogen or to have a remote sensor to see what is the level. But the liquid nitrogen, it is independent of energy in that sense.
Okay, thank you. So we have another question from Ananda, who is asking if you have experience with the cryopreservation of pin and Araucaria pollen, what is the moisture content?
I have not worked personally with pine pollen, but I know that a lot of forestry departments, they work with pine pollen for breeding purposes. And pine pollen tend to be orthodox. I suppose Araucaria pollen also. I'm not familiar with those species, but usually pine pollen is dried on like in a seed bank. It is dry at relative humidity in between 15 and 30, and then stored in the freezer at -20 or even -80. Because pine pollen, as far as I know, it is orthodox in storage behavior. So you can work with it like an orthodox seed.
Okay. Okay, thank you. Then we have another question from Ahmad, who's asking, how do you see the long-term sustainability of conserving plant genetic resources using cryopreservation? Can we avoid losing the longevity in any way?
No, the second question is, I think, liquid nitrogen. Alan is liquid nitrogen.
Liquid nitrogen anyway. Okay, sorry. So who can address this one? How do you see the long-term sustainability of conserving the genetic resources using cryopreservation? And if we can avoid losing the liquid nitrogen in any way. Okay, thanks.
Well, I can give my opinion, and Bart and Danny, you may have different. I don't know. Also, for me, it's the only way to go. The only technique that really, or method that really. Hello.
Yes, we can hear you. Go ahead, please.
The only method that really ensures the long-term conservation of plant genetic resources. In any other method, whatever, in vitro or orthodox seeds, intermediate seeds, at some point that the plant dies, or the ex-plant dies, and you have to regenerate it or multiply it. And so really long-term, I see there only cryopreservation. And can we avoid losing liquid nitrogen? Yes, there are tanks out there, high technology tanks that liquefy the evaporated nitrogen again. So these tanks you only have to fill up 1 or 2 times per year, but they're very expensive, and this is a new technology, so maybe not the first option to go for. But yes, it's possible.
Okay, thank you. So
please,
you
Bart and
want to add
Danny.
something, Daniel or Bart?
I fully agree with Rainer. I mean, there are some types of materials that, as far as we know, the only way to preserve them for the long term is in the temperatures provided by liquid nitrogen. And because of that, we need cryopreservation and we need liquid nitrogen. I suppose that as technology advances and allow us a more efficient use of the liquid nitrogen, and as Rainer said, some kind of recirculation, but liquid nitrogen evaporates because it warms up, and when evaporates, it expands the volume 100 times, so it is not an easy to handle gas. And yeah, I mean, you need liquid nitrogen, you need to provide it, but it is the same for any type of conservation we do ex situ, because if you have a conventional freezer, you need electricity. And for how long we can keep electricity? I mean, it's the same for liquid nitrogen. You need liquid nitrogen, you need to produce it and deliver. It is a matter of that you can continue doing that as long as you need it.
Okay, thank you very much, Daniel. So we have the question from Norma from Alliance Bioversity CIAT. She's asking, what about the fusion series tanks technology as alternative for area with difficult or lack of liquid nitrogen supply?
Well, yes, Norma, that's a good application of this tank, but I would not rely only on this option, because as it's good always to have let's say a backup tank next to your tank. If something goes wrong, you can move your material to the other tank. But what you're mentioning there is surely one of the best applications of this kind of tank.
Okay. So thank you very much to all. Thank you very much to our speaker for your excellent presentation today. So because now we are at the end of this session, so to all also for your active participation in today's webinar. So I would like to sincerely thank you for your presence. We look forward to see you tomorrow at 1 p.m. again, European time. And so please connect on time so that we continue this nice webinar series. Thank you very much for coming. Thank you. Bye-bye. See you tomorrow. Bye-bye.
Thank you. Bye.
Bye-bye.
Bye-bye.